Hello students. Welcome back to your favorite learning channel, Golgi Paathshaala. Today, we are going to study one of the most important and fascinating chapter of science, cell, the building block of life. Have you ever wondered, what is our body made of? How do plants grow? How does a wound heal automatically? And how can such tiny cells control the entire body? Well, today we are entering a magical microscopic world, a world that we cannot see with our naked eyes, but a world that is responsible for all life on Earth. From giant galaxies in space, to the tiniest microscopic structures, life exists everywhere. Every plant, every animal, every human being, including you, is alive because of tiny living units called cells. Today, we are going to enter a hidden microscopic world that exists inside every living organism. Your body may look solid from outside, but inside, trillions of tiny cells are constantly working. Whenever your wound heals, your heart beats, your eyes blink, or your hair grows, cells are silently performing incredible functions every second. So let us explore the amazing world of cells. In this video, we will understand the complete chapter in very easy language with deep explanation, including discovery of cells, microscope, cell membrane, osmosis and diffusion, cell wall, cell organelles, mitosis and meiosis, cell theory, important activities and experiments. So sit back comfortably, grab your notebook, and let's begin our exciting journey into the world of cells. Let us first understand what exactly a cell is. A cell is the basic structural and functional unit of life. Now what does that mean? It means that every living organism is made up of cells, and all life activities are performed by cells. Just like a building is made up of bricks, living organisms are made up of cells. Cells are extremely small, but they perform very important functions such as respiration, nutrition, growth, reproduction and excretion. Slowly, simple molecules combined and formed the first living cells. And that is why cells are called the building blocks of life. Just like bricks build a building, cells build every living organism. Some organisms are made up of only one cell. These are called unicellular organisms. Examples include amoeba, paramecium and bacteria. A single amoeba cell can eat food, move, breathe and reproduce all by itself. On the other hand, organisms like humans, plants and animals are made up of millions and trillions of cells. Such organisms are called multicellular organisms. Different cells in multicellular organisms perform different functions. For example, nerve cells carry messages, muscle cells help in movement and blood cells transport oxygen. Now let us learn how cells were discovered. In the year 1665, an English scientist named Robert Hooke observed a thin slice of cork under a microscope. Cork is the outer protective covering of tree bark. When Hooke looked through the microscope, he saw many tiny box-like compartments. These compartments looked similar to small rooms in a monastery. He named these tiny compartments cells. This discovery completely changed biology forever. However, the cork cells observed by Robert Hooke were dead cells. To study tiny cells, scientists use a microscope. A microscope magnifies objects and makes them appear larger. Important parts of a microscope include eyepiece, objective lens, stage, mirror, adjustment knobs. The specimen is placed on the stage. Light passes through the specimen and enlarged images are observed through the eyepiece. Microscopes help scientists study cells, bacteria and other microscopic organisms. Scientists later developed powerful electron microscopes that can even show structures at the nanometer level. With the development of modern microscopes, scientists were able to observe living cells and cell organelles in detail. Cells are extremely small in size. Different cells have different shapes and sizes depending on their functions. For example, nerve cells are long so they can carry messages over long distances. Red blood cells are circular and flexible so they can move easily through blood vessels. The ostrich egg is one of the largest single cells, while bacterial cells are among the smallest cells. The size of cells is usually measured in micrometers. Activity: estimating the size of a cell. In this activity, students estimate the size of an onion peel cell using a microscope. First, the diameter of the microscope's field of view is measured. Then the number of cells visible across that diameter is counted. This activity helps students understand how tiny cells really are. Every cell is surrounded by a thin protective covering called the cell membrane or plasma membrane. The cell membrane separates the contents of the cell from the external environment. It protects the cell and controls the movement of substances in and out of the cell. The cell membrane is selectively permeable. This means it allows only selected substances to pass through it. Activity: potato experiment. In this experiment, one potato piece is placed in plain water. Another is placed in concentrated salt solution. After some time, the potato in plain water swells. The potato in salt solution shrinks. Why does this happen? Because of a process called osmosis. Osmosis is the movement of water molecules from a region of higher water concentration to a region of lower water concentration through a selectively permeable membrane. Let us understand this with a real-life example. When raisins are soaked in water, they swell up. Why does this happen? Because water enters the raisin cells through osmosis. Similarly, when cucumber slices are sprinkled with salt, they release water and shrink. This happens because water moves out of the cells. Osmosis plays a very important role in plants and animals. Another important process is diffusion. Diffusion is the movement of particles from a region of higher concentration to a region of lower concentration. For example, when perfume is sprayed in one corner of a room, its smell gradually spreads throughout the room. This happens because perfume particles move from an area of high concentration to low concentration. Similarly, oxygen and carbon dioxide move in and out of cells through diffusion. Students often confuse diffusion and osmosis. Let's make it simple. Diffusion equals movement of particles. Osmosis equals movement of water only. And osmosis always occurs through a selectively permeable membrane. Cells behave differently in different solutions. In a hypertonic solution, water enters the cell and the cell swells. In a hypertonic solution, water leaves the cell and the cell shrinks. In an isotonic solution, there is no net movement of water and the cell remains normal. These conditions are very important for maintaining proper functioning of cells. Ever wondered what guards a microscopic cell? It's the cell membrane, an incredibly thin barrier made of lipids and proteins, structured just like a fluid mosaic. Picture a shifting bilayer of fat molecules, where water-loving heads face out and water-repelling tails hide inside. Because these molecules constantly move, flip, and rotate, the entire structure acts like a fluid. Meanwhile, embedded proteins act as literal gatekeepers, deciding exactly what gets to pass through. But while animal cells stop here, plants, fungi, and bacteria add an extra, tough outer shield called a cell wall, built purely for heavy-duty protection and structural support. Activity: onion and cheek peels. First, let's look at plant cells. Prepare a temporary slide using a thin onion peel or a Rio leaf, stain it with saffron, and secure it with a cover slip. Next, for animal cells, gently scrape the inside of your cheek with a clean swab, spread it onto a slide, and add a few drops of methylene blue stain. Under the microscope, look at the contrast. The onion cells are box-shaped and neatly organized, while the cheek cells are completely irregular and scattered. Now, let's push things further. Prepare fresh slides of both, add a 20% sugar solution, and wait 30 minutes. What happens? In the plant cells, the tough outer boundaries stay rigid, but the inner contents visibly shrink away. Meanwhile, the cheek cells, lacking that rigid outer wall, shrink down completely. Plant cells contain an additional rigid covering called the cell wall. The cell wall is made up of cellulose. It provides strength, protection and rigidity to the plant cell. The cell wall helps plants remain upright and prevents them from bursting when excess water enters the cell. Animal cells do not have a cell wall. That is why animal cells are usually flexible in shape. Now let's move inside the cell. A cell has three main parts: cell membrane, cytoplasm, nucleus. Inside the cytoplasm are many tiny structures called cell organelles. Each organelle performs a specific function. Inside the cell, many tiny specialized structures are present. These structures are called cell organelles. Each organelle performs a specific function just like different departments in a factory. Let us now study the important cell organelles one by one. Cells are broadly divided into two types: prokaryotic cells and eukaryotic cells. Prokaryotic cells are simple and small. They do not have a true nucleus or membrane-bound organelles. Bacteria are examples of prokaryotic cells. Eukaryotic cells are larger and more complex. They contain a true nucleus and membrane-bound organelles. Plant cells and animal cells are examples of eukaryotic cells. The nucleus is one of the most important organelles of the cell. It is usually present near the center of the cell. The nucleus controls all activities of the cell. That is why it is often called the brain or the control center of the cell. The nucleus contains genetic material called DNA. DNA carries hereditary information from parents to offspring. Characteristics such as eye color, hair type and height are controlled by genes present in DNA. The nucleus is surrounded by a nuclear membrane. Inside the nucleus, thread-like structures called chromosomes are present. Ribosomes are tiny structures that help in protein synthesis. Proteins are required for growth and repair of the body. That is why ribosomes are often called the protein factories of the cell. The endoplasmic reticulum, also called is a network of membranes present inside the cell. It helps in the transport of materials from one part of the cell to another. There are two types of endoplasmic reticulum. The rough endoplasmic reticulum contains ribosomes on its surface and helps in protein synthesis. The smooth endoplasmic reticulum does not contain ribosomes and helps in fat and hormone synthesis. The Golgi apparatus acts like the packaging and shipping center of the cell. It modifies proteins, packs them and transports them to different parts of the cell. The Golgi apparatus is made up of flattened membrane-bound sacs. You can think of the Golgi apparatus as a packaging and delivery center of the cell. Did you know the Golgi apparatus was hidden in plain sight? Until Italian scientist Camillo Golgi first spotted a strange, thread-like network inside the nerve cells of a barn owl. Because early microscopes couldn't show it clearly, many scientists actually doubted it even existed. It took decades, but the invention of the electron microscope finally proved he was right all along. Once its complex structure was clearly seen and confirmed, it was officially named the Golgi Apparatus, in his honor. Lysosomes are known as the cleanup system of the cell. Lysosomes contain digestive enzymes. They digest waste materials and damaged organelles. These enzymes help break down waste materials, damaged organelles and foreign particles. Sometimes, if a cell gets damaged badly, lysosomes may burst and digest the entire cell. That is why lysosomes are sometimes called suicide bags of the cell. And cut. That wraps up part 1 of our journey into cell, the building block of life. Let's take a quick breath and look at the massive ground we covered today. We started right at the beginning with the introduction and answered the ultimate question, what is a cell? From there, we explored the microscopic world of unicellular and multicellular organisms, and decoded the cell's outermost security guards, the cell membrane and the rigid cell wall. We also unlocked the secrets of cellular transport, mastering diffusion, osmosis, and how cells behave in hypertonic, isotonic, and hypertonic solutions. Finally, we took a deep dive inside the cell itself to study its parts and organelles. We compared prokaryotic and eukaryotic cells, and explored the power centers of the cell, nucleus, ribosomes, endoplasmic reticulum, Golgi apparatus, and those tiny recycling trucks, the lysosomes. That is a lot of incredible science packed into one video. But guess what? The cellular adventure isn't over yet. To see how the rest of this microscopic world works, including mitochondria, plastids, vacuoles, and how cells divide, you need to check out part 2. Until then, if this video helped you understand cells a little better, don't forget to like, share, and subscribe to Golu ki Paathshaala. Keep learning, keep exploring, and I'll see you in part 2.